first step to make some 149 media is you want to grab a 50 ml tube empty, and you're going to fill it up with the base, which is F12 1X, and you're going to fill it up 94% of your 15 ml total. So we have about 47 milliliters of our base F12. Once the base is in, the next step is to add our fetal bovine serum, or FBS, as well as our antibiotics. Over here we have pen strep, and it's going to be 1% of pen strep and 5% of the FBS serum. Now everything's just going to go back in the fridge. And then from there, our last three ingredients will be insulin, 1 milligram per milliliter, hydrocortisone, both of which can be found in the -20 freezer, as well as our heppies, which is used to form the buffer in the media. So our insulin is stored in the -20, however it is in the frozen form, so we will put it in the hot water bath. And in the meantime, we can continue adding our hydrocortisone. So for a 50 ml solution, we're going to add 25 microliters of 2 milligram per milliliter hydrocortisone. For our insulin, which is being warmed up, it's going to be 250 microliters of 1 milligram per milliliter insulin. And we'll just give it a few more seconds for it to warm up. Now that our insulin is thawed, we can go ahead and add that. And then our last ingredient, which we always want to make sure to add last, is our heppies. And the reason why we add it last is because it forms the buffer that the cells will be maintained in. And so for our Hepis, we're going to add 500 microliters of our 1 molar Hepi solution. So with all our ingredients together, we're just going to mix our 50 ml tube. And that's how we make some 149 media. Good. So once we have our complete media for some 149 prepared, we're going to begin by passaging the cells. And right before we start, we're just going to aliquot 5 mLs of our fresh media, and we're going to place it in a 15 ml tube to place in the hot water bath. From there, we're going to collect our ingredients we need to passage the cells, which include PBS as well as trypsin. So the clear solution is sterile PBS as well as our trypsin, which is a peptidase enzyme used to bring the cells into single cell suspension. So now that everything is set up, we can bring out our cells from the incubator. And they're currently at about 75% confluence. So again, when passaging cells, our first step is going to be to aspirate the media and do a quick PBS wash. So once the media is aspirated, we're going to add 5 mLs of PBS, and this is to get rid of any media residue so that our trypsin can work. Once our PBS has been washed through thoroughly, we're just going to aspirate the PBS. And at the same time, you want to make sure to work quickly to prevent the cells from drying. Our next step is going to be to add 2 mLs of trypsin. once again, the trypsin will work to detach the cells from each other as well as from the surface of the flask. And so we want to make sure it's distributed evenly throughout the flask. And because these are SUM 149 cells, we're going to incubate them at 37 degrees for about 7 minutes. Just going to start the timer for 7 minutes. So our 7 minutes are almost up, so we're going to collect our media from the hot water bath. And this media that's preheated will be used to neutralize our trypsin. And so we'll collect our flask from the incubator. So following a 7-minute incubation, we just want to make sure to tap the flask gently along the side. And this is to help the cells enter a single cell suspension. And so it is important to neutralize the trypsin as soon as we can because it is not healthy for the cells to remain in the cell. So we're just going to take our media, about 4 mils, and add it into our flask to neutralize the trypsin. And then we're going to waterfall the solution up and down to collect as many cells as we can. Once that is done, we're going to collect all our cells and place it into our 15 ml tube. So to continue maintaining the cell line, our goal is going to be to add fresh media into our flask, and we're going to add approximately 1/7 of what we have in here back into our flask, and the cells will continue to grow. So because the fresh media is cold, we're just going to add the media for now, and we're going to place it in the incubator. And after around 5 minutes, we will add our cells so that we can continue with cell culture. So I'm just adding about 20 milliliters of fresh media. So because the cells are collected in this 15 ml tube, we can continue to expand these cells back into the flask, or we can use these cells for future experiments. And yeah, that's it. We're ready to put our cells back in our flask with the warm media. We're just going to give the cells a good shake, just to make sure they're evenly distributed. And we're going to add 1/7 back. So because we have around 7 milliliters, we're just going to take 1 milliliter and add it back into our flask, which will then continue to grow. You always want to check on the microscope to make sure that the cells are actually there. And so can leave these cells in the incubator overnight, and that's how we expand and culture some 149 cells.